to all the students of 12th standard BPC, and today we are here on discussing the gunshot long answer type of questions from the chapter Respiration and Plants. So, in our previous class we have dealt with a short long answer type of questions from photosynthesis in higher plants and how we can connect learning these long answers to our NEET preparation and our EAP set as well. All the concepts that we are learning from this chapter, that is respiration and higher plants, 3 long answers are for sure to all the students who are attempting botany IPE exam. 3 long answers. Those are what? Glycolysis. First is what ma? Glycolysis, second is Krebs cycle, and third will be your electron transport system. These are the 3 that you have to learn for sure. Okay. This is important not just for IPE but also for your MCIT and your NEET exam. So, when you are learning this long answer, just if you highlight a bit on the bioenergetics, that will be helpful for the other 2 entrances as well. How you have to present this long answer? First you look at the question ma, explain the reactions of Krebs cycle. How many times it has appeared in your previous years of the IPE paper? See, in March 2024, February 2024, May 2022, June 2019, March 2019, March 2017, May 2016, these many papers you can observe the Krebs cycle question. That important question this is. So, here if you see, the Krebs cycle is a series of enzyme mediated cyclic reactions. In the beginning of the answer, any long answer it might be, the very first thing you have to do is you have to organize the question in a systematic way beta. You have to organize the question in a systematic way and present it accordingly. In the beginning though, first you have to give the introduction. The Krebs cycle is a series of enzyme mediated cyclic reactions that take place in the mitochondrial matrix. So, what happens after glycolysis that occurs in the cytoplasm? Just see here, once we will recall the previous story which has happened in the cell. If you consider this as a cell, inside cell cytoplasm, what happens ma? Inside the cell's cytoplasm, glycolysis occurs. After glycolysis, the pyruvate which is there, the pyruvate is directly sent to the mitochondrial matrix. Okay. The pyruvate is directly sent to where? The mitochondrial matrix. Fine, right. So, this is mitochondrial matrix, this is outer mitochondrial membrane, and this is inner mitochondrial membrane, and at the center this will be what? Intermembrane space. Clear, right ma? So, here in the cell's cytoplasm glycolysis occurs. After glycolysis, pyruvate which is produced, that pyruvate will be in mitochondrial matrix. Pyruvate which is produced in the mitochondrial matrix will start getting oxidized in oxidative decarboxylation, a step where pyruvate is first converted to acetyl-CoA. Okay. Pyruvate in the mitochondrial matrix, after glycolysis, if it is conducting aerobic respiration, first pyruvate is converted to what ma? Acetyl-CoA. This happens where? In the mitochondrial matrix by pyruvate dehydrogenase enzyme, or you can say pyruvate decarboxylase enzyme. In this step, 2 NADH are produced for 2 pyruvate converting to 2 acetyl-CoA will be releasing out what? 2 NADH here. And the enzyme is pyruvate dehydrogenase and pyruvate decarboxylase. This is how you can see, you can get the acetyl-CoA for Krebs cycle. Acetyl-CoA will begin the Krebs cycle. And what is that organic compound which is accepting the acetyl-CoA? Oxaloacetic acid. Oxaloacetic acid is a 4-carbon compound and acetyl-CoA is 2-carbon compound. Remember that. So, what happens after glycolysis which is in cytoplasm? Pyruvate, which is the end product of glycolysis, will be directly landed to mitochondrial matrix. In mitochondrial matrix, the Krebs cycle is operated. Krebs cycle is addressed as what? Enzyme mediated cyclic reactions which occurs. Location is very important. My examiner will be looking for these terms. Where is the reaction happening? Mitochondrial matrix. It is a central metabolic pathway in what type of respiration? Aerobic respiration. It is the central metabolic pathway. where the acetyl-CoA, that is acetyl group from acetyl-CoA, is completely oxidized to carbon dioxide in the Krebs cycle. Fine, right? With the release of the energy stored in the form of NADH, FADH2 and ATP. These are the energy rich molecules which are produced at the end of Krebs cycle inside mitochondrial matrix. So, after Krebs cycle, mitochondrial matrix will be rich with the energy components like NADH, FADH2 and ATP. This is the introductory line that you have to give, beta. Introductory statements that you have to produce. That is, it is an enzyme mediated cyclic reaction which occurs in mitochondrial matrix, and this is a central metabolic pathway of aerobic respiration. And at the end of Krebs cycle, what is that you are getting? That is NADH, FADH2 and ATP. This is what you have to produce in the introduction of Krebs cycle. Clear, right? Now we will look on to the diagrammatic representation of Krebs cycle. Very important. So, first I will explain you the complete Krebs cycle here itself, and then you can go for the theory. It is completely up to your comfort. Like if you want to deliver this question, first introduction, then diagram and then point wise description of each step. That is fine. Or first introduction about Krebs cycle and then point wise description of each step of Krebs cycle and last your drawing diagram. That is also fine. It is completely up to your comfort. But as a tutor, I will advise you to draw the diagram first, because this will help you, this flow chart will be helping you on easily analyzing each step. Easily you can remember each step when you are describing the answer. Okay, so I first advise you to draw the Krebs cycle and then you go describing each step of Krebs cycle. So here you see the very first step is condensation, wherein acetyl-CoA, which is 2-carbon compound, and oxaloacetate, which is a 4-carbon compound, will be condensed by the enzyme citrate synthase into citric acid. Okay, citric acid is 6-carbon, and then by the aconitase enzyme, this citric acid will be converted to isocitrate. Okay, this second step is isomerization. First step is condensation. Clear, right? Next, isocitrate will be decarboxylated to 5-carbon compound. Okay, 6-carbon compound is decarboxylated to 5-carbon compound. This third step is what, decarboxylation. Okay, this is decarboxylation, wherein 6-carbon is done to 5-carbon, that is alpha ketoglutaric acid, and here at the decarboxylation carbon dioxide is being released and NADH is reduced to what? NAD plus is reduced to what? NADH, and this is catalyzed by the enzyme isocitrate dehydrogenase. Then alpha ketoglutaric acid, which is 5-carbon compound, second decarboxylation will occur here. This is first decarboxylation, and here fourth step, this is second decarboxylation. This second decarboxylation, where 5-carbon compound, that is alpha ketoglutaric acid, is turned to 4-carbon succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase. Here also NAD plus is reduced to what, beta? NAD plus is reduced to NADH. Then succinyl-CoA is converted to succinate. At this step, substrate level phosphorylation will occur, SLP. Okay, this is substrate level phosphorylation, wherein from succinyl-CoA, when it is converting to succinate, from the substrate itself phosphate group is taken and GDP is converted to GTP, or you can also say ADP is converted to ATP. Next, succinate to fumarate. This is again 4 carbon compound, and here also it is 4 carbon compound. This is dehydrogenation reaction, wherein this is catalyzed by the enzyme succinate dehydrogenase. Sixth step, that is the enzyme is succinate dehydrogenase, where FAD plus is reduced to FADH2. Okay. Next, fumarate hydrolysis reaction. Seventh is what? Hydrolysis. Addition of water group by the enzyme fumarase. This turns to what? 4 carbon malate. Malate again dehydrogenation to oxaloacetate by the enzyme malate dehydrogenase. Here NAD plus will again get reduced to NADH. So, in this complete sequential reaction, 2 decarboxylation reactions will occur, that is from isocitrate to alpha ketoglutarate and from alpha ketoglutarate to succinyl CoA. One substrate level phosphorylation occurs, that is from succinyl CoA to succinate. 2 dehydrogenation reactions from succinate to fumarate and malate to oxaloacetate. Anyhow, you will remember the entire cycle. The entire cycle is important for IPE exam, but then at the time of your preparation, just highlight, just get focused where and how many decarboxylation reactions are happening. Here if you see beta, 2 decarboxylations, 2 dehydrogenations, 1 substrate level phosphorylation, 1 condensation reaction are happening. So, in a single Krebs cycle, if we have to calculate the bioenergetics, like how much amount of energy is produced into the mitochondrial matrix after the Krebs cycle, if you have to analyze in that way, firstly the cycle starts from acetyl CoA, right. So, a single acetyl CoA will continue the complete Krebs cycle, but if you have to calculate for 1 glucose will give rise to 2 pyruvate. 2 pyruvate will be turning to 2 acetyl CoA, but single acetyl CoA will complete 1 Krebs cycle. As such, for 2 acetyl CoA, 2 Krebs cycles will be completed. Okay, 2 acetyl CoA will conduct 2 Krebs cycles. Remember that beta. But for a single Krebs cycle, how much amount of NADH, how much amount of FADH2 and how much amount of ATP is released? That is the question though. So, you have to remember here 1 NADH and here NADH, and the last step, 8th step, also 1 NADH is produced. So, from a single Krebs cycle, 3 NADH are produced. 3 NADH will be released out, okay, from a single Krebs cycle. Okay. Fine. Next, how many FADH2? FADH2, if you see beta, 1 FADH2, that is from succinate to fumarate. So, it is 1 FADH2. Next, how many ATP or GTP? 1 ATP or GTP. And then how many SLPs are happening? 1 SLP over here. How many carbon dioxides are released? 2 carbon dioxides are released in the Krebs cycle. So, these are the energetics. These are the energetics from a single Krebs cycle. Okay, from a single Krebs cycle, this many compounds will be released. This is the amount of energy, that is 3 NADH, 1 FADH2 and 1 ATP. Clear, right? You can just go through the entire cycle, and if you are having any doubt, you can ask me beta. And here in the introduction, it will be very good if you highlight the synonyms as well. Like Krebs cycle, the name is given after the scientist Hans Krebs, who has discovered this. Okay, Hans Krebs has given out the Krebs cycle, who has worked out on flight muscles of pigeon. And then this is also termed as TCA cycle. This is also termed as TCA cycle, which is tricarboxylic acid cycle. Okay, as 3 carboxyl groups you can see here in the products, intermediary products. So, it is addressed as what? TCA cycle. And Krebs cycle after the scientist Hans Krebs. Okay, just go through the cycle. If you are having any doubt, you can just drop a message in the chat box. I will be explaining that. Yeah, if it is done, just let me know, ma. Any doubt anywhere, beta? If you just recall the cycle, it is enough. Okay, you can completely elaborate this. So if you go to the theory of Krebs cycle, first steps of Krebs cycle, formation of acetyl CoA. Okay, first is what, ma? Formation of acetyl CoA, that is pyruvate from glycolysis is converted to acetyl CoA by the enzyme pyruvate dehydrogenase. You can either give the enzyme name as pyruvate dehydrogenase or pyruvate decarboxylase, anything is fine, releasing out carbon dioxide and NADH. But this will not come under Krebs cycle, this comes under what? Link reaction. You can add on this under the title of link reaction. Okay. Now here you see condensation step wherein acetyl CoA along with oxaloacetic acid, this forms a 6 carbon citric acid, fine, right? And the enzyme here you see, the enzyme is what? Citrate synthase over here, which is condensing acetyl CoA and OAA forming citric acid. Next is isomerization wherein citric acid by the enzyme cis aconitase is converted to isocitric acid by the enzyme what? Aconitase. When you are describing each and every step of Krebs cycle, it is very important to tell about the reactants and products, type of reaction and enzyme, that is it. Okay, because examiner will be looking for the name of the reaction, raw materials, end products and enzyme, enough. This you can write it comfortably, easily when you first present the cycle and then you start on explaining the cycle. That will be easy for you, ma'am. And then you see after isomerization, fourth step is what? First oxidation or first decarboxylation. Here if you see isocitric acid is turned to oxalosuccinic acid, which is reduced to what? Alpha ketoglutaric acid, that is 5 carbon compound. Okay. Here the enzyme is isocitrate dehydrogenase and the product formed is what? 1-NADH. Next you see second oxidative decarboxylation wherein alpha ketoglutaric acid, which is 5 carbon, is turned to 4 carbon succinyl CoA. So here which is the intermediary step, you can eliminate this beta. If you want to write, you can write. If you want to eliminate, you can eliminate, because first isocitric acid is turning to oxalosuccinic and then alpha ketoglutaric. This is the flow of description. It is comfortable. If you are comfortable with the intermediary products, you can write. If not, you can eliminate, directly give out the product name. Fine. Next enzyme over here is alpha ketoglutarate dehydrogenase complex. Product formed is what? 1-NADH again at the end of oxidative decarboxylation. Next, sixth step if you see, this is substrate level phosphorylation or cleavage, wherein succinyl CoA is converted to succinic acid and coenzyme. This will generate 1 ATP or 1 GTP by the enzyme what? Succinate thiokinase. Next, third oxidation, succinic acid to fumaric acid by what enzyme? Succinate dehydrogenase. This is the enzyme. Product formed is 1-FADH2. Fine, right. Next, hydration. Fumaric acid to malic acid, which is 4 carbon, and the enzyme over here is fumarase. Fourth oxidation, that is malic acid to oxaloacetic acid, where the enzyme that is involved is malate dehydrogenase. Fine, right. And the product formed is 1-NADH. Fine, right. So here thus oxaloacetic acid, which is 4 carbon, is regenerated, completing the cycle. Okay, regeneration of OAA is done, which is the primary acceptor of acetyl CoA. Okay, regeneration of OA is done. This is the primary acceptor of what? Acetyl CoA. Now the energetics for one turn of Krebs cycle. Energetics also you can calculate easily, you can analyze easily when you first present the cycle itself. So here you see beta, energetics of Krebs cycle, first 3 NADH, 1 NADH is equal to how much ATP? 3 ATP, right? So, in a single cycle 3 NADH are produced from where to where? Oxalosuccinic acid to alpha ketoglutaric acid, alpha ketoglutaric acid to succinyl-CoA, and at the step where malate to oxaloacetate is formed, that is the last step of Krebs cycle. At these 3 areas 3 NADH are produced, wherein 1 NADH is equal to 3 ATP. Find right. 1 NADH is equal to 3 ATP. So, here 3 NADH is equal to 9 ATP. Next, 1 FADH. 1 FADH is equal to 2 ATP. This is via electron transport system. Find right. 1 ATP is produced at substrate level phosphorylation, that is from succinyl-CoA to succinate by the enzyme succinate thiokinase. Next is total. In total, how many ATP are produced, beta? 12 ATP equivalents are produced at the end. Since one glucose molecule produces 2 acetyl-CoA, one glucose will produce 2 acetyl-CoA. So, the cycle turns twice per glucose. I told you already, right, when I am explaining. The cycle will turn twice per glucose. This will yield how much? 6 NADH. 6 NADH means 18 ATP. 2 FADH2 is equal to 4 ATP and 2 ATP indirect. As such, grand total from a single glucose molecule only in Krebs cycle, if you have to count for single glucose molecule through Krebs cycle, the energy produced is 24 ATP, which is bulk amount of energy. This complete energy is stored in mitochondrial matrix. Mitochondrial matrix. Find right, ma? So, these are the energetics of it. Next, significance of Krebs cycle. Central hub of metabolism, that is amphibolic pathway, both catabolic and anabolic pathways. Okay, that is Krebs cycle, because in a Krebs cycle the intermediates which are produced like alpha ketoglutaric acid might be succinic acid, oxalosuccinic acid, these are the intermediates for the biosynthetic pathways of many secondary metabolites. As such, Krebs cycle stands as an amphibolic pathway which includes both catabolic and anabolic reactions. Clear, right? So, in the energetics part of you have to produce from a single Krebs cycle how much amount of energy is produced, and from a single glucose molecule from Krebs cycle how much amount of energy is produced, and at the end you have to mention the significance of Krebs cycle. This is the way you have to present this question, ma. Okay. This produces coenzymes. Coenzymes are produced like NADH and FADH2 that drive ATP synthesis via oxidative phosphorylation. Next, this provides intermediates for amino acid, nucleotide and lipid biosynthesis. That is why this is called as what? Amphibolic pathway. This completes the oxidation of carbohydrates, fats, proteins into carbon dioxide and water. Clear? Any doubt anywhere, ma? No, right? So, first in the introduction, discovery of the Krebs cycle, major points of Krebs cycle, and then draw the complete Krebs cycle, elaborate each step of Krebs cycle. In the elaboration of Krebs cycle, what you have to highlight, ma? Name of the reaction, raw materials, end products and the enzyme of that particular reaction. And lastly, bioenergetics, energetics of a single Krebs cycle and energetics of one glucose molecule where the Krebs cycle turns twice. As such, 24 ATP is the total energy that is produced. And lastly, you have to give out the significance of Krebs cycle. This is the way you have to present this question to get the fullest marks. Okay? And in this way, if you learn this answer, this will be helpful for EAP set and NEET as well. Next, you see electron transport system. This is the last question. This is the last question to be targeted, beta. Okay, this is the last question from this chapter. So, look at here, oxygen is critical for aerobic respiration. Explain its role with respect to ETS, electron transport system. Here you see ma electron transport system. First you need to again give a proper introduction about this electron transport system, that in aerobic pathway this is the last and final step to be conducted. So, in aerobic pathway, aerobic respiration involves complete oxidation of glucose. Okay, this is where the complete oxidation of glucose, which is turning into what? Carbon dioxide and water with the release of energy, that is ATP, simplest form of energy in a cell, or the direct form of energy that is utilized for metabolism is ATP. Next, after glycolysis and Krebs cycle, the reduced coenzymes NADH plus H plus and FADH2 will carry high energy electrons to ETS, electron transport system or electron transport chain. These are located in the inner mitochondrial membrane. This point you will learn in cell biology also. All the enzymes, protein channels, electrons, whichever are required for electron transport system or electron transport chain are present in the inner mitochondrial membrane. The final utilization of oxygen occurs at the terminal stage. So, the ultimate electron acceptor here in the electron transport system is oxygen. Fine, right? This is the basic description that you have to give for ETS. Don't be that student who prepares for IPE separately, who prepares for MSET separately and who prepares for NEET separately. Nah, that doesn't work. Just when you are going through the long answer type of questions or short answer type or VSAQs, just go through each question in objective perspective, like where is it happening, how is it happening, what are the proper enzymes, what is the bioenergetics. If you learn in this way, that will be useful for your IPE, EAP set and NEET. You have to go in a combination. Okay, if you just blankly read these statements, remember it, memorize it, ratta mark it, and then if you write it 100 times, memorize it, vomit it in the IPE paper, after that you will again sit for MSET and NEET, that doesn't work. Okay, be a smart student who will cover the complete preparation in a single stretch. Okay, so to all the 11th and 12th students, if you are with me daily, botany syllabus will be covered completely for IPE in coordination with your EAP set and NEET. Fine, right? So, here you see first for EAP set or NEET or IPE, how you have to exercise electron transport system. Look into this clearly. Here also, after the introduction of ETS, where is it happening exactly in the inner mitochondrial membrane, and then you have to draw the proper diagram of electron transport system. See. So, here first if I have to present, here you see ma, careful. Here you see after glycolysis in mitochondrial matrix, first you in this cartoon diagram you just analyze the mitochondrial structure clearly. This is outer mitochondrial membrane, inner mitochondrial membrane, this is intermembrane space and this will be mitochondrial matrix. Clear till here no? In mitochondrial matrix, after Krebs cycle, let us consider for 1 glucose. After oxidative decarboxylation, 2 NADH are produced. Fine, right? Here I will write 2 NADH after oxidative decarboxylation. And in TCA cycle, total 6 NADH, this I am saying for 1 glucose. Fine, right? And how many FADH2? 2 FADH2 and 2 ATP. Now, these reducing molecules, energy rich molecules NADH and FADH2 will be oxidized completely in ETS. So, all the protein channels that are present in the inner mitochondrial membrane. Fine, right? Inner mitochondrial membrane, that is cristae, all the protein channels are present. As such, to operate electron transport system, here now NADH, FADH2 everything is present in the mitochondrial matrix. This is mitochondrial matrix beta, mitochondrial matrix, and this is inner mitochondrial membrane, okay, and this will be what, just a second, intermembrane space. Fine, right? Mitochondrial matrix, inner mitochondrial membrane, intermembrane space. Where is electron transport system operated? In the inner mitochondrial membrane. So what happens, you just see. The complete process is operated by 4 complexes. Complex 1 is known as NADH dehydrogenase. Complex 1 is NADH dehydrogenase. And complex 2 will be succinate dehydrogenase. Complex 3 is cytochrome BC complex. Complex 4 is cytochrome C oxidase. Fine, right? And last complex you can call it as ATP synthase complex, which is complex 5. Here you see, ma, yeah, first complex 1, that is NADH dehydrogenase, NADH plus H plus, which is in the mitochondrial matrix. Okay, NADH plus H plus, which is in the mitochondrial matrix. This will be oxidized, releasing out NAD plus 4 protons plus 2 electrons. Fine, right? Plus 4 protons plus 2 electrons. 4 protons from mitochondrial matrix will be directly pumped to intermembrane space. 4 protons will be directly pumped to intermembrane space. And 2 electrons which are there, they will be accepted by the protein molecule, that is flavin mononucleotide FMN from complex 1. Okay, 2 electrons will be accepted by FMN, that is flavin mononucleotide from complex 1. 4 protons will be directly pumped from the mitochondrial matrix to intermembrane space. So here from flavin mononucleotide, that is FMN, 2 electrons are again submitted to what? Ferrous sulfur complex. Okay, and here electrons are then submitted to a mobile electron carrier. Mobile electron carrier. What is a mobile electron carrier? The carrier which carries the electrons between 2 complexes, from one complex to the other, that is the mobile electron carrier, that is ubiquinone. Accepting the electrons, this will be reduced to hydroubiquinone. Okay. Now complex 2 you see. Complex 2 is placed after 3 here to explain you that complex 2 is not transmembrane protein channel beta. Here you can see that this is transpanning 2 sides of the membrane, complex 1. Okay, if you just recall the protein channels, you have 3 types: integral proteins, transmembrane proteins and extrinsic proteins. So here first complex is what? That is transmembrane protein, which transfers the electrons from mitochondrial matrix to intermembrane space. But here the complex 3 if you see, sorry, complex 2 if you see, this is not transpanning both sides of the membrane, only it is attached to inner mitochondrial membrane, is attached to only mitochondrial matrix. So here you see succinate dehydrogenase, that is FADH2 is reduced to again FAD plus 4H plus 2 electrons. So here again both protons and electrons are accepted by FAD plus, that is flavin adenine dinucleotide, as this cannot pump protons to intermembrane space directly. And from here you see, from here FAD plus is pumping 2 electrons to ferrous sulfur complex, then to the mobile electron carrier, that is ubiquinone. This will pass out 2 electrons to and this will get reduced to what? hydroubiquinone. From here, both the electrons and protons of complex 2 and the electrons from complex 1 are given to complex 3, that is cytochrome Bc complex. So, here in cytochrome Bc complex, both electrons from both the ends are given to cytochrome B, protons are submitted to the intermembrane space. On total, 4 protons from complex 3 are again reduced to intermembrane space. One thing to be highlighted, all the complexes will work parallelly. No complex will wait the work completion of other complex, then it will operate. Now, all the complexes here will work cooperatively. Okay. So, here you see from complex 1 4 protons, from 3 4 again, because complex 2 is submitting both electrons and protons to complex 3 and complex 1 is giving out the electrons to complex 3. As such, protons are submitted from complex 3 to intermembrane space. From here, again there is a mobile electron carrier which is carrying out the electrons from 3rd complex to 4th complex. Till here everything is clear na beta? Till here though it is clear, no? Okay. Hindi medium. This class is completely in English, ma. Later we will be starting for Hindi as well. But as of now though, this is completely for English. This will be in English only. The medium of this class is English, ma. In the next class we will be starting that. Fine, right beta? Till here if you are having any doubt you can ask me. Yeah. So, here you have complex 1, 2 and 3. Till here everything is clear, no? Here comes another electron carrier, mobile electron carrier, which is from the intermembrane space facing to from the inner mitochondrial membrane, which is facing towards intermembrane space. Okay. Again a mobile electron carrier which is from inner mitochondrial membrane facing to intermembrane space. Fine, right? So, here cytochrome C, this is giving electrons to copper molecule. Okay. And then cytochrome A, A3 and then to copper. So, here in complex 4 if you see cytochrome A, A3 and copper molecules. Like how you have flavin mononucleotide, FAD and here cytochrome B. And here in both all the 3 complexes, one thing is common if you observe, ferrous sulfur complex. But in complex 4 instead of ferrous sulfur complex, you have copper molecules along with cytochrome A. As such, from here the ultimate electron acceptor is oxygen atom, which is waiting since a very long time for electrons. Okay. So, here oxygen, once after getting the electrons, it will combine with 2 protons which are in the mitochondrial matrix and a water molecule will be released over here. If you look at the end of 4th complex, in the mitochondrial matrix, all the protons whichever are there, they are pumped in sequence to the intermembrane space. Yes or no? After pumping to the intermembrane space, the electron is being submitted to oxygen, whichever minimal protons are there, they are combining with oxygen and they are forming what? Water molecules. And then 2 protons from here again pumped to intermembrane space. So, if you see here, there is an electrochemical gradient which is created in the intermembrane space. Okay. From the inner mitochondrial membrane, in between mitochondrial matrix and intermembrane space, there is a proton gradient or electrochemical gradient is created, wherein more protons are on the intermembrane space, less protons are on the mitochondrial matrix. Now, these protons should be pumped back to the mitochondrial matrix to maintain that electrochemical gradient. So, here if you see, there is a pump which is operated, that pump is known as F0 F1 complex beta. Okay, this is known as F0 F1 complex. Okay, these are also known as oxysomes or Fernandez-Morgan particles as well. Okay, which is F0 F1 complex. This is also known as ATP synthase. Okay, so from here when the protons are being pumped, ADP and inorganic phosphate which is there in the mitochondrial matrix is converted to the energy rich molecule ATP. So, at the end here you can see ATP and water molecule is formed, right. As such, ATP molecules are formed from NADH and FADH2. ATP is called as what? Powerhouse. Fine, right? Energy currency of the cell. Okay, ATP is called as what? Energy currency of the cell. Why ATP is called as energy currency of the cell? Because a cell for all its metabolic activities will use the energy in the form of ATP itself. Clear, right, ma? So, clear with the complete electron transport system. If this is clear only, we can go to the proper theoretical explanation of this question. So, in the beginning what you have to give out? You have to properly frame out the introduction of electron transport system, draw the diagram, highlight 4 complexes, and you have to mention each protein molecule of each complex. Specially highlight the mobile electron carriers, and lastly electrochemical gradient, that is F0 F1 pump. Clear till here? If you are having any doubt, you can just ask me, beta. Clear, right? Now we will go with the theory. So you see complex 1, that is NADH dehydrogenase, ubiquinone oxidoreductase also you can call it as. If you are comfortable with this, you can mention. If not, in your answer this also will go fine. Okay, complex 1 NADH dehydrogenase. This accepts electrons from NADH plus H plus oxidizing back to NAD plus, which happens where? In the mitochondrial matrix. This transfers electrons to ubiquinone, coenzyme A, and reducing to ubiquinol or hydroubiquinol. Next, this pumps 4 protons from mitochondrial matrix to intermembrane space. This contributes to the formation of proton gradient, which is necessary for what? ATP synthesis. Fine, right? Next you see succinate dehydrogenase. This accepts electrons from FADH2. Okay, this accepts electrons from FADH2 generated during the conversion of succinate to fumarate in the Krebs cycle. Next, this transfers electrons directly to ubiquinone without any proton pumping. This is the sequential description, okay, of each complex. Remember that. If you are unable to maintain the same points also, no problem, ma. Just look into the explanation which I have just given with the diagram of electron transport system, and with proper terminology explain in your own terms, it will be accepted. Okay, but present the answer in a systematic way. Unlike complex 1, it does not span the entire inner membrane, provides an alternate entry point of electrons into ETS, but yields less ATP since only H plus are pumped from complex 2. Okay. Next, complex 3. Fine. Complex 3. Here you see complex 3. Yeah. Cytochrome BCS complex, which is also known as ubiquinone cytochrome C oxidoreductase. Ubiquinone cytochrome C oxidoreductase accepts electrons. So here there is a question, ma'am, you are too fast. Nothing like that, ma. I am on the base tempo only. If you are not comfortable with the speed or accuracy, you can pause and listen. There is no problem. Okay. Accepts electrons from reduced ubiquinol and transfers them to cytochrome C. This is the mobile electron carrier. Cytochrome C here is what? Mobile electron carrier. This pumps 4 protons into intermembrane space per pair of electrons. This contains cytochrome B and C1 along with ferrous sulfur complex. Okay. Acts as crucial middle link connecting ubiquinone with cytochrome C, maintaining the electron flow properly. Next, cytochrome 4, that is cytochrome C oxidase. This accepts electrons from cytochrome C and then passes them to molecular oxygen. Oxygen is reduced to water molecule by combining both electrons and protons. This pumps 2 protons into the intermembrane space. This is the final step of ETS. What all we have discussed, that only is being explained beta. And last, that is complex 5, which is ATP synthase and F0 F1 complex. This will utilize the proton gradient which is created by complex 1, 3 and 4. Protons will flow back into the mitochondrial matrix through what? F0 channel. This flow rotates F1 subunit, catalyzing the phosphorylation of ADP plus inorganic phosphate and ATP. Here, if you mention this point, you will get an add-on, like where ATP is formed actually. ATP is formed in mitochondrial matrix. Next, each full rotation of ATP synthase will produce 3 ATP molecules. Clear, right? Yeah. Next, you see, ma. Complex 5. ATP synthase, that is F0 F1 complex. This complete explanation is done. Each full rotation of ATP synthase will produce 2 ATP molecules. It is not 3 beta, it will produce 2 ATP molecules. So, this table is very important. Name of each complex, name of each complex, electron source. For complex 1 it is NADH, for complex 2 it is FADH2, 3 it is ubiquinol, and 4 it is cytochrome C, and 5 it is generated through the proton gradient. Electron destination, here for the first and third complexes the mobile electron carrier is ubiquinol, third complex it is cytochrome C, and then it is oxygen. Fine. Lastly, for ATP synthase the acceptor is ADP and inorganic phosphate. Proton pumps, first it is 4 protons, and from complex 3 it is 4, and then from complex 4 it is 2. Protons are pumped to intermembrane space, this you can mention. Fine, right, intermembrane space. This is the complete description of the answer. Clear, right, ma? Next. Table is very, very important. This table, if you present, it will be impressive, ma. Yeah. So, here if you see once again the electron transport system, how you have to present this answer. First ETS, then proper diagram for that, and each complex you explain in detail. At the end you present this table which will be impressive. That is it. This is how you have to go with the answer. So, from respiration in plants, 3 questions you have to highlight beta. One is glycolysis, second will be Krebs cycle or TCA cycle, last is your electron transport system. With this, respiration in plants chapter is done. In our coming class, we will discuss your short long answer type of questions from another chapter of your 12th BPC syllabus. Till then, stay tuned and thank you.